Hi, everyone. Thanks for joining. We'll get started in just a couple minutes here and give everyone a chance to sign into the Zoom, get settled in. In the meantime, if you haven't done so already, you can actually sign up for a Biorender account following the link that's on the screen right now. And if you do have an account, feel free to log in as well. That way you can follow along with us. If you have any questions, this is a really easy place to get some answers. And if you have any questions, then I'm just going to also pop the link into the chat here just so that everyone has it for easy access. I know that, oftentimes trying to copy and paste a link from a presentation is not the easiest. So I'm just going to drop that into the link, drop that link into the chat here as well. So again, if you don't have a Biorender account yet, that link will take you to create your own account. And if you do have one, again, feel free to sign in. And that way you can sort of follow along as well with the tips and the discussion that we're going to have. And we are a couple minutes past the hour now, so I'm going to go ahead and get started here. So welcome everyone to our webinar today, which is Top Tips for Designing and Delivering Great Presentations. We're going to spend the majority of the time focused on presentation figures, but we'll also cover some of the less thought of aspects of presentations as well. So it'll be a bit of a demo of Biorender, but I also wanted to share some general presentation tips if you ever did need to use other tools like PowerPoint or Keynote. And to quickly introduce myself, my name's Tim, and I'm a Science Communication and Customer Success Manager at Biorender. I'm a scientist by training. I did my undergrad in biochemistry before getting a PhD in pharmacology and therapeutics. And it was during my time in research where I experienced many of the challenges that all of you likely face on a day-to-day basis when you're going to create figures for your work. And that's why I joined Biorender, because I wanted to help scientists avoid the same problems and struggles that I had to go through and show them that there's an easier way to create figures that accurately represent their work. And just a few housekeeping items before we jump into things. Again, if you haven't done so already, you can sign up for your account, and I'll pop that link into the chat one more time just so that everyone has it. And if you're just joining us as well and you do have a Biorender account, feel free to sign in and follow along as well. Just a couple of housekeeping items here as well. First, if you do have any questions, you can ask them in the Q&A section in the Zoom window. When you do, remember to address the question to both panelists and attendees so that everyone can see. And then you can use the chat to say hi or chat with other members of the audience if you'd like. I'm joined by my colleague Mayette, who will be helping me answer questions that come in, so she'll be keeping an eye on both the Q&A and the chat. As a favor to the both of us, if you can put your questions into the Q&A, it'll help us a lot, just because if it is in the chat and there's a lot of activity, sometimes the question can get buried, and we just want to make sure that we're able to answer anything that you have on your minds. Now, we don't have a dedicated Q&A session, so if there's anything that you want to learn more about or anything is unclear throughout the session, you don't have to wait until the very end. Feel free to ask whenever that question pops into your mind, and we're happy to dive into it with you together. And before we dive into it, I just wanted to get to know all of you just a little bit better here. I'm going to launch a quick poll, just asking what your experience level is using Biorender and what you normally use Biorender for. So I just launched the poll. You should see it pop up on your screens soon. If you don't mind taking just a couple seconds to answer, that'd be very helpful. Awesome. Seeing a lot of responses coming in, which is great. So it looks like a lot of folks have at least heard of Biorender before. Some folks that are a little bit newer. And as expected, it looks like a lot of people are using Biorender to create presentation figures, which makes sense since you're all here with me today. And it also looks like a number of people use it to describe protocols and workflows. Definitely good things for us to keep in mind as well as we look to plan sort of these future webinars to help help scientists with their work. So thank you everyone for answering here. All right, so for those of you who are newer to Biorender, it's a graphics making tool, and it was developed because our co-founder Shizuoki realized during her time as medical illustrator that many scientists and labs don't have the budget nor the resources to visually translate their science. And this was really the spark that drove the founding of BioRender, because we wanted to give everyone the tools and the practical knowledge to visually communicate their research and allow everyone to clearly convey their message visually. And here are just some of the common ways that we see BioRender being used. For those of you who are power users or veterans, you probably already know this, but you could use it to create figures across your entire scientific journey, starting all the way from the ideation and brainstorming phases, all the way up to your publications, your patents, posters, and hopefully even more in the future. And of course, for today, our focus will be on presentations. And as scientists, I'm sure everyone knows presenting our work is an important part of what we do, whether it's to our own team, to other departments, at conferences, or presenting our project progress to leadership. It's a critical part in making sure that our work is understood and to make sure that we're making well-informed decisions regarding the direction that we want to take our research in. And the challenge here is that the people that we present to often have too much to do and too little time to do it in. And what this means is that we have a limited amount of time to make sure that the people that we present to our audience can clearly understand our message. And that's what the tips that we're going to cover today during the session are going to address. And that's how to get your message across as efficiently as possible to your audience. So let's get started here. And here's an agenda of what we're going to talk about. So the session is broken up into roughly 3 parts. We'll first do a quick review of hardware and software. And then we're going to look at some design tips for when you're creating your slide decks as a whole. And then we're going to get a little bit more granular and talk about some design tips for when you're designing your slide figures specifically. And again, you'll notice that there isn't a dedicated Q&A. So if you do have any questions throughout, feel free to throw them into the Q&A or the chat. We'll make sure to get to your question, and we do appreciate the engagement. It loves seeing the gears turning in your head to see how you can kind of incorporate those tips into your work. So feel free to ask anything that comes to your mind. All right, so let's get started here with a quick review of hardware and software that can be really helpful to engage your audience. And the really big thing here is regarding audio quality. And so with many of us meeting regularly over Zoom and presenting virtually, we might not always stop and think about the equipment that we're using to present. And so again, audio quality from the audience's point of view can really make or break your presentation. And so here's sort of a rough ranking of audio quality for different types of microphones. So we have AirPods at the very end, we have our laptop, some gaming headsets, standalone mics, as well as maybe a headset. And what might be surprising for everyone are where gaming headsets sort of fall in the spectrum. And while gaming headsets have they give really great sound because it allows you to hear all those little details in a game, they don't always have the best microphone quality because that's not what they're designed for, and it's just something to be aware of. And similarly, things like AirPods, for example, or other similar headphones, these are again optimized to give really good sound, but not always optimized for their microphone quality. And what I want to do right now is actually just run a quick test to kind of highlight the difference between my laptop's microphone and then the headset that I'm using. So what I'm going to do, actually, I'm going to switch over to my MacBook, to my MacBook's microphone. Let's take a look here and see how that works. And right now I'm using my my MacBook now, and you might be able to hear like very quickly the difference in that sound quality. It might sound a little bit more echoey, a little bit more distant because my mouth is further away from my microphone, so there's more room for the sound to bounce. And exactly, you can hear the echo already. And I'm in a room that has carpet, so that helps a little. But imagine being in an office that might have hardwood with concrete walls. That echo is going to be even more pronounced. And if you imagine having to listen to this kind of quality for extended periods of time, it's not going to be the best experience. And so what I'm going to do, I'm going to switch back to my microphone right now. Hopefully you can hear that difference right away. So it's going to sound a little bit more lush. The sound doesn't bounce as much, mainly also because like, the microphone is closer to my mouth as well. Um, and I'm not advocating for any specific brand or type of microphone. It's really, you're looking for something that was designed for teleconferencing. So like the headset that I'm using is just the one that's on the screen right here, but really you're just looking for something that's optimized for microphone quality. And again, the reason for this is that when we're virtual, the audio quality becomes more and more important, especially as you need to present for longer. If you imagine having to listen to me talk using my laptop microphone for the whole hour, it's not going to be a great experience. I mean, my voice aside, it's probably better to hear from the headset as opposed to the laptop. Um, And really, the message that I want everyone to take away here is to maybe ask a friend or a colleague to test your audio, even if it just means recording yourself speaking with different microphones and then playing it back for yourself to see what it's like. You can get a very good sense of like how your audience hears you. And also, don't be afraid to let your colleagues know if their audio quality isn't great, because it really does take away from the message that they're trying to deliver. And again, with everyone having so little time, you want to make sure you remove as many obstacles or barriers from your audience understanding your message. And audio quality would be one of those first things that you can easily address. All right, so now that we've kind of gotten that out of the way with regards to the hardware, let's jump right into the next category here, which is about design tips for overall slide decks. And it's definitely hard to give tips here because every presentation is going to be a little bit different, right? If you think about it, all the content that you want to show will differ from presentation to presentation. The length of your talk is going to be a little bit different. But I do think that there are some overarching tips that I think are applicable to any sort of presentation. And before we get into, what we should be doing for our slide decks, let's quickly talk about things that we shouldn't do. And so this slide here sort of has all of like the do nots all kind of lumped into one slide. And so the first is to avoid gradient background. So you can see right now we have a radial gradient that sort of starts off sort of lighter in the middle and gets darker as we move to the edges of the screen. And the reason we want to avoid this is one, it's difficult to do tastefully and it makes things difficult to read. And the reason it makes it difficult to read is because it has very inconsistent legibility. You can see that as we start from the edge here, this bullet point plus like the creates word is a little bit clearer than when we get to legibility. And so we want to have a more solid color background so that the legibility across your entire slide is going to be more consistent. The second is to avoid drop shadows. So this is a little bit more on the distracting side because it creates sort of like a 3D effect, which then draws your attention towards those shadows and maybe away from sort of that main message that you have on your slide. And so drop shadows are something to avoid as well. And reading through the slide here, you can see that, you're reading, you're kind of noticing those shadows, you're paying attention to that. And with focus, you can treat focus almost like a currency. So if I'm spending some on drop shadows, I'm taking it away from a more critical area that we could be focusing on. Whoops. Sorry about that. Just accidentally scrolling through my mouse pad here. But what you want to also do is avoid harsh colors, like this bright yellow font that we've used for the title here. Again, it's more distracting. And when it comes to designing your slides as well, you actually want to reserve bright colors for things that you truly want to highlight because it makes it stand out from everything else that's on your slide here. And so it's not that you shouldn't use color, it's just to limit the amount of bright or very intense colors that you're using so that when you do use them, it really jumps off the page to your audience. As well, you want to avoid decorative bullet points. And so you can kind of see some of those bullet points here. And the reason for this is that it creates a little bit more of a dated feel. And so for when you're creating more of a modern, timeless slide deck, stick to more basic bullet points, sort of like just a standard circle or a dash. Those work perfectly fine. As well, you want to avoid serif fonts and focus on using sans serif fonts. And I actually have some examples here to highlight this. And so here in this GIF, we have an example of one side that has that's using serif font and one side that's using sans serif font. And the reason that you want to focus on using sans serif font is that it helps your deck look cleaner and a little bit more modern. And it's not to say that there isn't a time and place to use serif font itself. Serif font is actually is really helpful for when you're reading through large bodies of text, like papers or textbooks, because then what happens is that each word is more like a picture than individual letters. So it helps you read sentences a little bit faster. However, when you are presenting a figure, usually you want to limit the amount of text that's on your slide as well. And so using Arial is going to create like an easier to read font for your audience. And you can see even when we compare the two that one does look a little bit more modern compared to the other. And when you're going to build your slide deck, and I have an example here, one thing that you want to keep in mind is to use a consistent feel and look for your deck. And so I have sort of an example slide template that I've created. And so flipping through here, you can actually see that we have different layouts throughout this template here. But what you'll notice is that all of them are very consistent. They share those kind of same color blocks. They share similar fonts as well as organization. And so the easy way to get this consistent look actually is to just go into the theme builder. PowerPoint and Keynote also have similar functionality. It's just really about how you get there. For Google Slides, it's in the theme builder. And here you can actually make changes to that master slide, which will then apply to all of your subsequent levels. And this is, again, a really easy way for us to create a very cohesive slide deck that we can use in our presentations. And of course, I'm sure that many of you will have branded templates that are provided by your organization. And those are fantastic to use because it really help give that uniform look to your deck. And it just looks a lot more professional when you're going to present your work. One tip that I do have for everyone here is actually to make your last slide and your first slide match. And so the reason for that is it forms really nice bookends for your presentation. You know that, this is the start of my presentation, and this is the end of my presentation. They look very similar. And with this last slide as well, really take advantage of it. We might not always notice this, but we spend a lot of time on that last slide. Just think of all the seminars that you've been in, all the presentations. After we go through all the content, when we get to the Q&A and the discussion, often that last slide is just up on the screen. And so to make sure that you're kind of maximizing the opportunity that you have with that last slide, you can do things like include contact information or maybe some relevant information that you really want your audience to walk away with. You can add some kudos or shout outs to people who have helped you with your project and things like that. And the key point here is to make sure that you're utilizing that last slide effectively because your audience will spend a lot of time looking at it. And one last tip that I have for everyone here, you can see sort of this like scrolling slide on the page right now. And this is the highlight that there will be times when you're going to have a slide that just have a bunch of different logos, maybe from collaborators, from other organizations that have contributed to the work, and you're going to need to find logos to represent them. And the problem is that it's not always easy to find logos that have a transparent background. So you might end up with like a mix of files, some with transparent background, some with not. And rather than having to find a transparent background for every single one of these files, what you can do instead is actually just put a white background behind everything, which then removes sort of like that cloud that surrounds the logo. And again, it just saves you that time of having to find the appropriate file for every single logo that you need for your deck. Does anyone have any questions so far about how to build your deck or anything else that I've talked about so far? Let's take a quick look into the chat and the Q&A. It looks like we're pretty clear right now. But again, if you do have any questions, feel free to pop them in there. We want to keep this as engaging as possible. If there are things that you want to see more of, let me know, and I'm happy to get into that more with you. All right. So continuing on here with creating your deck, sometimes what you might be, whoops, not this one here. This is the slide that I want. what you might be tempted to do is actually just take the figure that you're using for a paper or for another project and then just sticking it directly into your presentation. And this is something that I'm guilty of as well. You know, I have a pre-made figure, it's ready to go, and I'm just going to pop it into my presentation. But instead, now that I know better, what we recommend actually is to split it up. And this is mainly for legibility purposes. It again makes it like each individual panel a little bit easier to read. You have more legible font size, or you can make your font bigger. And it just makes it really easier for your audience to kind of see what's happening within the figure. You can even see that like right now, just comparing on the left that I have like, you know, a 6-panel figure. If I were to put that directly into a single slide, that's going to be pretty compressed. But if I break each of those slides into their own separate slides, it gives me a lot more room to work with, and it allows my audience to, again, focus on that one panel that I'm discussing at the moment. And it is something that it's easy to forget, especially right now. If you do a lot of meetings virtually, all of us have a dedicated screen. But when you go to present, you know, in person in larger conference rooms, we should always be cognizant of the size of your figure when you're presenting. And so there's something for everyone to keep in mind here. All right. And now that we've sort of covered some of those general tips for presentations and decks, I want to get more specifically into like the figure making portion of the session today. And I do see a couple questions that have come in here. Answer the one in the chat first. Someone's saying that it's not recommended to use bullet points in a PowerPoint presentation. Yeah, happy to comment on that here. I think bullets have a place in the presentations, mainly because it allows you to very clearly list like the different items. For example, here on my slide, you can see that we have like these 3 sections that we're covering, right? If we imagine removing the bullet points, it becomes a little bit harder to delineate which like the three sessions apart from each other. So I like using the bullets because it then clearly indicates like this is one item, this is a second item, this is a third item. However, I do think that there should be a limit to the amount of bullet points that you're using. Where I recommend using bullet points typically is if you have like a list of things that you want to present, because then it just clearly denotes each item. William, does that kind of help with the question that you have in mind? Awesome. And yeah, and so what I want to do right now is actually just hop into BioRender to kind of show you some tips that you could use when you're creating your slide deck, or for the figures for your presentation, sorry. And I'm going to start from the very beginning here, just to make sure that for those who are a little bit newer to the platform, that we all sort of start on that same footing. So when you first sign into your account here, you're going to land in your gallery page, and this is where you're going to find all of the illustrations that you've created before, as well as all of the illustrations that have been shared with you. And so here on the left, you can create some folders that help you keep you organized. You can create it for different projects or different things that you're working on. And then these files and folders that you create, these will be private to only yourself unless you decide to share them with others. And down below here, this is where you're going to find all the files and folders that have been shared with you. So essentially here in your gallery page, this is where you're going to find all the files that you have access to. And so for us, I'm going to hop back into the slide deck that I prepared for today. I'm just going to hop into this into my file here, which opens up in the editor. And to give everyone just sort of a quick overview and refresher for those that you're a little bit more familiar with the platform, here in the editor, this is where you can make like changes to your illustrations, and sort of like the key tabs that you're going to be working through are here on the left-hand side. I'm just going to use a couple of these for as an example. One of the main ones that you're going to be working on are likely going to be the icons. And so I'll use a new slide here to kind of highlight what we're doing here. And so here in the icon library, this is where you're going to find all these pre-made images that you can use to build your illustration. Wood signs, yeah, that we do have We do have plant icons. Let's take a look here. And so this can work into our examples as well. And so where we can do it, we just like either search for it. So maybe we can go into species. We have plant species, and then we can kind of review some of these icons that you might find useful for your illustration. What I'd recommend as well is that as you're browsing through all of these, as you're browsing through the icon library, if there are things that you think that we should add to our to the library itself, feel free to make a suggestion for it. Like, it's really helpful for us to understand, where we can continue to grow our library, maybe areas of research that are up and coming. So if there are suggestions that you want to see, you can definitely submit a suggestion here. And so you can review to see what sort of plant icons we have. And if you need to, you can also search for icons as well. And so if we type in wood, for example, it does look like we're a little bit shy here that we have a generic tree. We might be able to find more specific things by searching a specific type of tree like oak. So this might help with what you have in mind here. And what you'll notice that as we go through with all of these icons, everything that you see here, to use them in your work, all you need to do is just drag them out of the library onto your canvas to start using it in your work. And then from here, you can do a lot of things like edit the appearance of the icon by changing its color, changing some of its other properties to get it looking exactly the way that you want for your image. And next, in terms of building blocks for your illustration, what you also encounter are the brushes. Now, the brushes are these dynamic pieces that allow you to create sort of customized structures for your illustration as well. And so as you scroll through the brushes tab, you'll see that we have brushes for membranes, we have nucleic acids, vesicles, bacterial membranes, cell brushes. And the really cool thing about all of these brushes is it allows you to create, again, this customized structure. So I'm going to use this endothelial brush layer just as an example. And what you notice right away is that it has these blue lines along with these nodes that are scattered between. And this allows you to kind of create like these customized structures where your icons will then follow the path of the line that you create. And this allows you to create very custom structures for all of your work. And so definitely explore the brushes themselves. I know for folks that are in more of the cell biology side of things, phospholipid bilayers would have been a nightmare to create by hand. So if you haven't done so already, we do have phospholipid layers along with all the subunits already shown, so that all you need to do is just bring that out onto your canvas, and then you can start just adjusting it, and you'll see that we automatically add each of those subunits in for you, and you don't have to worry about adjusting it or copy and pasting, hundreds of these units together. And if you imagine sort of like those icons and the brushes being like individual building blocks as you go to construct your figure, then the templates are fully formed illustrations or structures that we give for you to use. And so here in the templates tab, you'll see all the different categories of templates that are available to you. And again, just like with the icons, feel free to browse through each of these individual categories if you're just looking to see what's available, or if you have something more specific in mind, you can again search for it as well. So for example, if you're looking for a specific maybe IMD pathway, you can type that into the search bar, and here you can see that pop up. And to use it, just click on the template, either choose to add it to your illustration or replace your entire illustration altogether, and then that template will be loaded up. And the really cool thing here about the templates is that they're made up of all these individual pieces that are all available within the BioRender library, meaning all of these templates are fully editable illustrations. If you need to, you can remove things from the illustration. So let's say in my example today, I'm giving a talk. I don't actually need steps 7 and 8. It's a little bit distracting. So I'm going to just remove it from my illustration here. So I'm just going to delete that. And I can just tuck in that title a little bit, and then also adjust the canvas so that we remove some of that excess white space. And now, when we go to present our figure, we get our audience focusing on just like this key portion of that illustration. And when you go and design these slide decks, personally, I really like the templates because they're really easy for you to like give the background information to your for your talk, especially if you need to catch your audience up or if they're maybe from like a different field or a different department. The templates are a great way to find some of that background information to include in your deck without having to find something off of Google and then needing to creatively crop it or obscure information. Using the templates, you can actually just remove all the extraneous work and have your audience focus on just like those key portions of the illustration that you want. So again, going back to that, what we talked about earlier, how we want to lower the activation energy for our readers or our audience to absorb information by having just like the critical parts of the key bits of information on our template. And by removing that extraneous information, it really helps to make sure that our audience's focus is on the right things. All right, so now that all of us are sort of on like that same or a little bit more of even footing of like how BioRender works, how you might be able to create figures with it, what I want to do is now actually just get into the tips for when you're designing those slide figures specifically. And so the tips that we chose here might not, or the examples that I'm using, sorry, might not necessarily cover your exact field of study, but the tips I think are generally applicable and you can apply them to your own figures. So you can kind of think of this as like a cross-discipline affair. Now, the first tip that I have for everyone is always check for your contrast and accessibility. So one of the more common mistakes that we see with figure making is that the figure isn't legible because elements are fading into each other. And I have an example here, which kind of highlights this. And so, as you go through your figures, you're changing colors, maybe either by using the color presets, or you're even going into individual layers as well to change like the color. And you'll notice that with the icons, you actually have a lot of this flexibility as well. If you haven't tried it already, you can actually hop into the different layers, often for cells or a subset of these icons. And here you can actually get very creative with your colors. So you can either use this color customizer to get very specific with the color that you want, or you can choose from the color defaults down below. Or if your organization has very specific colors that they want you to use, anywhere that you can customize color, you can also add in hex and RGB codes as well to get like that specific look that you need for your illustration. Now, the moral of the story here is that you have a lot of control over how you want your colors to look. But the thing to keep in mind is that regardless of the color you pick, you want to make sure that there's enough contrast between your foreground elements and your background elements. And the best way to do that is to actually counterintuitively convert your figure into black and white to take a look at the contrast. And so if we can do that here in BioRender, just go to View Canvas and Grayscale, and you'll see that we strip away all of the color. And the reason this works is because color has three properties, and the two that we're looking at right now are hue and value. And by going into Grayscale, we actually remove hue from the equation, and we're looking specifically at color value. And when you have colors that are very similar in value, they tend to blend in together into each other a little bit more. And when you convert into a grayscale and you see this effect where you have those two elements sort of blend in or they sort of disappear a little bit, that's our cue to go back in and change the darkness or the lightness of these elements to have them stand out a bit more. And for example, if I toggle back with color, at first, the DNA looks like, it stands out decently from the nucleus, but when I go to strip the color away from it, you'll see that it sort of just fades into it a little bit more. And what we can do instead is let's try and make that DNA a little bit darker to kind of create a bigger difference between the color value of my foreground and my background. And so with my DNA selected, I'm just going to turn up the value here so it actually becomes a lot darker. And we'll notice that when we're in grayscale, those particles faded in a little bit. So I'm going to repeat the same thing with those particles as well. I'm just going to make it a little bit darker so that everyone can kind of see like the effect. And now, once I go into grayscale, you'll see that the my DNA and those particles don't actually disappear into the background anymore. And so this means that when I do have the color available, it becomes a lot more clear. And so in a sense, we've kind of fixed up that cell. And if we look at our cancer cell on the right-hand side, we see something very similar. When we have color, the label is semi-legible. When I remove color, it definitely fades a little bit more. And so what we can do is repeat the same thing. I'm going to select that text. I'm going to go over to color, and then we're going to turn it into a black. So it really just stands out here. And now you'll see now my cancer cell label very clearly stands out against the nucleus itself. But the nucleus can be tricky to adjust for contrast because naturally it stains darker than the rest of the cell. But even though it's dark, we have to layer in a lot of elements like DNA or label, or we have to add arrows and proteins. And there's a practical limit to how dark we can make something in the foreground. And so instead of making the foreground continuously darker, we can actually approach this problem from the other direction as well, and we can turn up the transparency or lighten the background. And so what we can do is select that cancer cell. And so here in the editing panel, we have a property for transparency. And we can actually turn up the transparency here. And you can see that as we do it, that text starts to stand out more and more from my cell itself. And so we can have that label really pop and really stand out from the rest of our cell. So after that, if we go into grayscale one more time here, you can see that everything that all these changes that we made stand out quite nicely, and it's a lot easier for my audience to read. I see a couple questions here. How do you use the color picker? Yeah, that's a great question. And so to use the color picker, all you need to do is select the elements or the icon that you want to change the color for. And then from here, if they have individual layers, you can actually go in to change those layers separately. Or if it is just like the overall, if it's like a single unit, what we can do is actually overlay our icon with a certain color. So we click on this plus sign here. Here in the color picker, we can either use this. Oh, sorry. And by color picker, I think you're explicitly referring to the color picker itself. Essentially, it's a sampler. So if you click on color picker, you'll see that as I hover the eyedropper over my different elements, that there's a little field on my top left here that shows like what color that I'm about to select. And once I do that, it will actually just change like that color specifically. So I think I have my T cell sort of all selected. And so if I select from my particles, you see that my entire cell has now been kind of overlaid with that color. Marty, does that help with the question that you have in mind there? Awesome. And Christine, I see that Mayed is answering your question as well. But tips on picking colors that won't get totally washed out on projectors or on a screen, something that you can keep in mind. And I think Mayed also has sort of that described as well. Again, you might want to try with maybe like a darker background with lighter text, as well as maybe some brighter colors that stand out from my background. Or if you're using a light or a white background, you can use darker colors to kind of help you make things stand out a bit more. So if you imagine on a projector right now, my background will be very white, but because I have like these darker colors for like my text and my icons, it's going to stand out from those from like that background. And even if it is washed out, it'll still stand out from my from the rest of my illustration. And so with the colors washing out, you can kind of think of it as almost like a contrast issue as well. And so if I were to go into grayscale, for example, here, instead of just checking the foreground that I'm looking at is my illustration, and my background is going to be like the slide background here. And so you can kind of apply those same principles that we're just looking at. And what we can do is just make sure that my foreground is dark enough. So if I am using a light background, even if things get washed out a little bit more, there is still sufficient contrast between those two elements. So Christine, hopefully that helps with your question there. And on the topic of legibility, what I want to talk a little bit about here is about font sizes. And this kind of builds, whoops, let me put some color back in here. And it sort of builds on like that topic of legibility that we were just discussing already. And like we talked about earlier, when you're at home or you're presenting remotely, you can probably get away with a slightly smaller text size because everyone has their own monitor or their own computer. And if things get a little bit small, you can always just lean in a little bit. But for in-person meetings, or if you're in a larger auditorium or even just a conference room, that's not going to work as well. It's gonna, even if someone leans into the screen a little bit, there's only so much that they can lean in without falling out of their chair. And more importantly, not just for the concept of font size, what you want is actually an appropriate font size to to slide ratio. And what I mean by that here, so if I go into present, you'll notice that all this text is relatively legible. Some of it's a little bit smaller, but you can probably get away with this. And what's actually really surprising, this is actually size 8 font. So it's actually very small. And what you're looking at then is actually looking at like that font size to canvas size ratio. And so the reason that this works is because the ratio of our font, or sorry, of my canvas here is actually quite small. The canvas itself is only 10 inches wide by 6 inches high. So relatively small. Even if I blow that up into the screen, that scale stays relatively similar. However, if I make this canvas a lot larger, we're just going to make this a lot bigger here. All of a sudden, when I go to present, that same size 8 font becomes a lot smaller. And so when you go to present your slides, keep that in mind. It's not necessarily that absolute font size, but it's what you want, but that ratio to the font size against the size of your deck. And so to fix this up, super easy, what we can do is actually just go in here. We'll zoom back in so you can see what I'm doing. Is actually select all of the text. So I'm just going to select these one at a time here. And then what we can do is just blow this up a little bit. And so right now everything is font size 8. We can maybe push that to 14. And if we go back into like that original sort of example where I'm just going to make this slide deck nice and big, what you'll see when I go to present is that all of a sudden that text becomes more legible because again, it's about the ratio of the font size against your canvas. And obviously right now, by changing my font size, a lot of that's going to just throw all of my text boxes out of whack. So if you do see that, your text boxes are kind of skewed or pushed because the font changes. What you can also do is actually leverage some of those alignment and distribution tools that we have in BioRender. So for example, this is going to be a very rough example here. I'm just going to select these boxes. Maybe I want all of them to be aligned against like the top edge of the box. What I can do is actually go select my 4 elements, go up to align, and then here I have the option of aligning against different edges. And so if I want all of the top of my boxes to line up, I can select this option here. And this will then move all my text boxes upwards so that it all lines up perfectly. So kind of easy way to address that here. And I think it'll save a lot of time of having to manually readjust like those text boxes. Of course, when you do have different size text boxes, you want to fix that as well. But in terms of realigning all the boxes, using that is going to be a very quick way for us to push everything together. And one tip that I do want to share with everyone here is that when you go to present in person, especially for larger conferences or you have a very big audience, what you can do is actually go into the room ahead of time, maybe load up your slide deck, put it onto the projector on the big screen, and then go to the back of the room that you're in. And so this way, you can make sure that you are able to read everything that's on the back from the back of the room, and because that's going to mimic what the audience will see. And so this way, as long as you can read it, that means your audience is able to read it as well. So just an easy sort of quality control check that you can do to make sure that your font is legible. And are there any questions here about legibility or color and contrast? I see. Look, just reading the chat here, looks like pretty good. Thanks so much, Maya, for fielding all of those questions. And thank you everyone for asking. Again, we'd love to see like the engagement from everyone to just let us know that things are making sense at the very least. All right. And so the next tip that I want to talk about here is when you need to go and create a more complex figure. And I wanted to, and then like from this complex figure, you want to highlight a small portion of it. And so for example, I have my stereotactic device on my page right now, and maybe I want to zoom into the mouse and I want to make that a little bit clearer. So one way to do this is by trying to make this illustration extremely big to try and get that mouse into sort of a more visible or like a larger, it takes up more space. But it's not really practical, right? If I make this bigger just so that I can see the mouse, the rest of my stereotactic device gets enormous. And now you can see that it's falling out of my slide. It's kind of out of, it doesn't sit in my slide deck very well. And it just simply making the figure bigger is not going to be a viable option. So instead, what I'm going to do here is actually use the crop tool to help us kind of create like this step-by-step zoom in process. Oh, and I see a question here before we dive into the cropping feature. The perfect ratio between the size of the title and the text in the slide. That's a really good question. I think typically what I've seen is to have titles, at least for posters, are like a 96-point slide. And then using like a slightly smaller, maybe like a 32 for the body. However, I don't know if there's like a one size fits all sort of solution. I think it really depends on what you're trying to emphasize in your slide. Definitely like if there are, let's say, a heading 1, like your title, and then a heading 2, heading 3, you want those to get progressively smaller. Or if you're going from, let's say, like a title to just like body or normal text, the gap would be a little bit wider. I think some of that will be a bit of an art where you can play around to see like what helps you create that hierarchy, because that font size can really play a role in it. So definitely your title will always be the largest font, and then your heading 1 is going to be a step down from that, your second heading, your third, and then to your body of text. And so it really depends on like the hierarchy that you're trying to create. Catalina, awesome. Glad that's helpful. But yeah, going back to sort of this cropping here, if I wanted to make this mouse a little bit more legible, what I'm actually going to do is first make this smaller, which is a little bit counterintuitive, right? If I want to zoom into something, why am I making this smaller? And so the reason for this is that it's going to give me a little bit more room to work with, and you can kind of see what happens as we put this illustration together. And so next, I'm going to create a duplicate of my illustration. So just by pressing and holding Alt or Option if you're on a Mac and dragging, we're able to create like this duplicate image. And then with this duplicate image, I'm going to make this one bigger. And if I wanted to focus on the mouse, I'm going to make the mouse like the size that I want it to be. So I'm just going to make it a little bit bigger here. And then I'm going to take advantage of the crop tool. So if I go up to crop on the top toolbar, I had the option of cropping as a square or a circle or a custom shape if I wanted. In this case, I'm just going to stick with a circle. I like it because it looks like a magnifying glass. You can definitely choose whichever one you prefer. I just like using the circle. And so what I'm going to do after, I'm going to trim away everything that I don't want. So I'm going to make this like about the same size as my mouse, kind of expand this a little bit. And then now when I go to finish, when I click outside the crop, you can see that I've trimmed away everything now. And now all of a sudden my mouse becomes a little bit clearer. And then what I can do is even look for maybe a zoom icon to link those two together. Let's grab, maybe I like this one. So we can grab this one here, and I'm going to fit that lens to the same size as my mouse. Just going to shrink this a little bit. You can kind of play around with it to make sure like everything fits appropriately, or alternatively, you can also, the easier way might actually be to adjust the image underneath. So I'm going to go ahead and just tuck those in a little. And then I can actually take these tails and then just point it at my mouse. So you know what? This is my mouse here. Bring that in. Now, all of a sudden, I still have my stereotaxic device in place, but I also have a clear view of the mouse. And then if I want to take it even one step further, and let's say I want to get that really blow up the size of that brain, I'm going to repeat the same process that I just did. So I'm going to make a duplicate of this, and then I'm going to make this nice and big so I can see what I'm doing. And then I'm going to edit my crop this time. And now I'm actually just going to make it so the crop focuses just on the mouse's head. Whoops. I'm going to move this here. And then just make it a little bit bigger, like so. And now when I click out, I can progressively make this larger. And what I can do if I wanted to make sure that these two sort of zooms are equal, what I'm going to do is actually just select this image. Let's see how big this is. It's about 1.9. So I'm going to do the same thing here, scale and rotate. It's going to unlock the aspect ratio, make this 1.9 as well. Make this 1.9. And now we have like the same size circle here. And to kind of address Becky's question that's in the Q&A, there isn't actually a loss if you're working with biorender images. There isn't any loss in resolution because we do work with vector-based files. So you can see that even with like these progressive zooms, even when we make it much larger, we still have everything is still very clear because of that. I think Maya might have some suggestions in her answer as well. But it shouldn't be a concern if you are working within biorender because everything is a vector-based image, and so you can make things a lot larger without needing to worry about that degradation. And again, you can kind of see from here we made this mouse progressively larger, and it's still very very sharp. And yeah, just to kind of complete this image, I'm just gonna make my just kind of added my zoom lens here as well. Again, just adjusting this. And the really cool thing about being able to sort of use this more step-by-step zoom process is that it's a much gentler way of helping your audience kind of walk through your illustration. And so rather than make just one big image and then like having something small, having the step-by-step zoom kind of helps you build that story, right? And so it's a lot easier for your audience to follow. And so like the theme of what we've been talking about today, the easier your illustration is for your audience to follow along with, the easier it is for them to absorb that information. And lastly, what I want to talk about here on like that topic of story, when you go to present your figures, oftentimes you're going to have very complex illustrations to present. And when you have this, what if I go to talk about this illustration or this pathway here, when I'm talking about SARS-CoV-2 binding, my audience might actually be looking here down below at like this pathway, or maybe they're looking here at like which proteins are inhibited or like the drug structure that I was using. So they're not completely focused on what you're talking about. So instead, to make sure that again your audience is your audience's attention is where you want it to be, what we can do is actually make this illustration come together in a step-by-step format. And I think a lot of us already kind of do this intuitively. So we'll have, let's say, like multiple slides all showing like the completed figure. And then what we're going to do is make each of those elements appear individually. And so here, this is going to be my final figure. It all comes together here. But maybe in my third slide, I want them to just focus on the proteins that are being inhibited. And I can repeat the same thing. And then here, delete all of these. And I want to talk about the binding aspect. And now when I go to present my figure, so as I'm talking through, my SARS-CoV-2 virus binds my ACE2 receptor, it gets taken into my cell, which then triggers a number of different steps. And then if I inhibit it, these are like the proteins that by adding my drug, I can actually inhibit these proteins from being synthesized. And then the structure of my drug is here. And so by walking through your audience through the story, it allows them to follow everything and it's almost like a smoother flow as opposed to showing them everything all at once and having them try and absorb everything like all at one time. You're kind of breaking it down into like smaller bite-sized pieces for your audience. And another trick as well for storytelling, something that you might not have considered yet, is to actually have your elements fade in and out. And so previously, the tips that we're talking, the what we did here was actually to make things just appear. So we just start off with that one piece and have each piece progressively show up. But another way that you can do things is actually have it almost like a spotlight. So using this example of some protein analysis, you can see that our story kind of splits into 3 chapters. We have our first chapter looking at protein preparation, then we have a PTM scan analysis, and then last we have some spectrometry, as well as some computational analysis of our protein. And again, like before, if we were to present the slide all on its own, it's a lot of information to digest. And if I'm talking about step 1, someone might be looking at my data in step 3 and might not be the most helpful. So what I can do is actually leverage that transparency feature in BioRender. So I'm going to drag a box over my 2 steps first. Actually, I'm going to make a couple of duplicates here. So duplicate 3 times. All right. So in this first step, when I first start off talking, I want to make sure that my audience is focused on chapter 1. So I'm going to select 2 and 3, and I'm just going to turn up the transparency maybe to about 80%. And then after, in my second part, I want to talk about the second chapter. So I'm going to turn this to 80, turn this to 80. And lastly, down below, I want to focus on step 3. And now when I go to present my illustration, you can first start off with like a general overview of your protocol. And then afterwards, you can talk about the protein preparation step in more detail. And the audience now knows to focus on that highlighted step. Once you're done discussing how you prepare your protein, you can move on to PTM scan analysis. And right away, even when I flip between the 2 slides, you might have noticed that your eyes will have instantly went from the protein preparation to step 2. So kind of cool way to kind of like guide your audience through the illustration as well. And then lastly, when I go to step 3, we can discuss like the analysis. And the reason why we want to have the spotlight is because it keeps all 3 chapters sort of in context with each other, while also allowing our audience to focus on like the portion that we want them to focus on. Are there any questions about how to tell a story or how we can guide our audience through our illustrations? Oh, see a question here. Can you use BioRender to draw chemical and drug structures? That's a really good question. Right now, there isn't. If we don't have anything that's like ChemDraw. What we do have in the icon library, though, are a series of different chemistry elements. So if you have like certain molecules in here that you can draw from, you might be able to pull some basic structures here. Alternatively, you can put it together as more of like a grouped icon where you have like a series of text boxes and lines that you then connect together. Adele, that is a really good suggestion. I'll bring that back to my team as well to let them know that having an ability to draw chemical structures would be very handy. Christine, great question. Thoughts on a progress bar timeline at the bottom of the slide? I think that's a great idea. I think it's again, a lot of it is just imagining how you can guide your audience through your illustrations. So I think a progress bar is a great idea. So you let them know that like, hey, this is the beginning. This is like about halfway through. This is like the end of my story. It really helps like just keeps your audience engaged in that sense. Becky, it's suggested to break down busy slides into individual ones, but time constraints and complex data sets make this really difficult. The transparency suggestion is great. Are there any others? Actually, so the transparency one is a really good one. Obviously, there's the appearance. However, I think one myth that I want to address here is having 1 slide per minute. So I think it partly depends on how much information that you have on the slide. And so I was definitely on the 1 slide per minute camp for a long time until a colleague of mine actually was talking that if you want to keep your audience engaged, it's actually better to have more slides that you spend less amount of time on, and then just flip through the slides a little bit more regularly. And then so in that way, you actually spend less than a minute a slide. And this allows you to kind of break apart that larger 1 minute slide and maybe spend like 15 to 20 seconds for like the slightly smaller slides. So that could be like an easy way to kind of break apart some more complex information. And I think like even just adding, let's say like you can use color to help like highlight things. So transparency is one aspect. If you use color to guide, so if you have everything a little bit more on like a pastel color, and then you have bright colors to highlight what you want to talk about, that's another way of sort of guiding your audience through your illustration as well. Hopefully that kind of gives you some ideas and jogs and then like gives you some inspiration there. If you have any ideas that you want to share as well, like we'd love to hear it. And I know we're almost at time here. So what I wanted to do actually was just to launch one final poll, just asking you how you find how informative that you found this webinar. So I'm just going to quickly launch this here. And if you don't mind just taking a couple seconds to answer before you head out, that'd be greatly appreciated. And to wrap up here, what I wanted to do is really just to thank everyone for taking the time to join us. Like, we love the questions. Hopefully everyone got some better ideas on how they can create more effective slide decks and figures for their slides. Can really appreciate everyone taking the time to join me. And to kind of take this full circle, you can see that, we're on that last slide right now. We're saying those these final Q&A, this discussion. And here you can see some information that I've included. So if you do want to bring BioRender to your organization, feel free to contact support@biorender.com. As well, if you have any questions that we didn't get to today or anything that pops up later on, support or this help button down below are great ways to ask those questions. So again, thank you so much, everyone, for taking the time to join us, for your support of BioRender. I hope all of you found this seminar informative, and I'm excited to see the figures that you create.